The mechanisms by which sexual dimorphism develops has not been fully understood despite its significance in ecological diversification. Sexual dimorphism is the differences between members of the same species related to the gender of the organism from the same population. Common examples of dimorphisms are a lion's mane or a rooster's tail. To address this fundamental biological question, this study utilized the sexually dimorphic horns in the dung beetle species Onthophagus taurus. In O. taurus, just like other beetles, their horns play an important ecological role in males, i.e., competition for females. Furthermore, the insect sex determination gene doublesex has been shown to be involved in the horn development. Therefore, this species provides an ideal opportunity to study the genetic underpinnings of sexual trait development. The focus of this study is on genes intersex and transformer2 and their significance in the development of O. taurus horns in relation to doublesex. Transformer2 is assumed to be a regulator of the sex-specific alternative splicing, a process during gene expression that results in a single gene encoding for multiple proteins of double-sex pre-mRNA in female Drosophila or fruit flies, while remaining inactive in males. Intersex in Drosophila is proposed to be a transcriptional cofactor of doublesex, but like transformer2, it is inactive in male flies. The function of intersex and transformer2 in beetles and their horns are not currently known. Thus, to examine their function in the development in general, and to understand the function of those genes in the horn development, systematic knockdowns of intersex and transformer2 will be implemented by RNAi. So far, RNA has been extracted using triazole reagent, and cDNA was synthesized from the RNA by reverse transcription. PCR was then performed to amplify the protein coding region of intersex, and the product was cloned in the plasmid vector. The plasmid was then purified for sequencing. After isolating the gene-specific region, double-stranded RNA was synthesized using the purified plasmid. Results from previous experiments in our lab have shown that knocking down intersex creates male traits in females, like horn development. Future methods include isolating transformer2 and injecting RNAi to the third instar larvae. Since intersex appears to be transcribed in the same mRNA size based on our present results, we expect that the protein intersex is not produced via alternative splicing. Rather, it's possible that it's the physical interaction with the female-specific transcription factor doublesex that mediates differentiation. While the expression pattern of transformer2 in O. taurus remains unknown, it can be assumed that based on previous research, transformer2 is indeed responsible for splicing double-sex pre-mRNA into its male and female specific forms. Knockdowns of this gene in Tribolium castaneum, which is a red flower beetle, led to the presence of the male-specific form of double-sex and the development of pseudomales. Therefore, if found to be similar in O. taurus, females may develop the pseudomale traits such as horn development after double-stranded RNA injection. Our future directions include observing potential missplicings of double-sex female and double-sex male proteins by running PCR using injected beetles after knocking down transformer2. To physically observe the interactions between double-sex and intersex, we will stain tissues using antibodies against the double-sex protein after injection.